Very good, folks. Okay, so let's get going. I've got an extensive presentation this morning because ultimately this invitation allowed me to review 30 years of my life, and try to do this in 2 hours is difficult. Thank you very much for the invitation. Okay, I've known Omer's connection to the Society of North American Scholars for a couple of years. And I was actually tickled when he invited me to do this. I look at this very much as trying to teach a potential new generation of individuals in something that I've been working on for a long time, which is novel. And this is in part background, in part research methodology, in part how do you apply this, what's good, what's bad, and life experience, all packed into one presentation. So at very beginning, I always like to look at definitions. And you will see this morning, I'm not going to read slides verbatim. You have access to these slides. So if there's anything that's of interest to you, by all means, take a look at that. And if you've got questions, pretty easy to get in contact with me. So about 30 years ago, my interest was entirely leadership. And we defined something called leadership neuroscience because at a conference, I had serendipitously had an opportunity of speaking to somebody that worked a lot with teenagers on attention deficit disorder. And I was very fascinated with the technology that they used to actually change the human brain profile to correct issues like ADD and ADHD. And I just asked a rhetorical question. I says, I wonder if we could actually change the behaviors of leaders that I know that were actually very good behaviors. And the guy says, I don't know. That's why I came to your conference on leadership. So that got me to talk to this gentleman and to learn what they were doing. And we just did what we call the back of a napkin experiment. And I found that, hey, I could actually predict stuff with this guy's stuff. And I was very skeptical. But we did define leadership neuroscience as kind of a science, okay? Taking a look at the brain and trying to explain how the brain, or what is the role of the brain in terms of emotions, your thoughts, your decision-making processes, and ultimately your actions as a leader. And then when I decided to make this one of my research goals, I had a dual role. Assessing leadership, yeah, you're good at this or you're not good at this. But my passion was developing leadership, using new methodologies, because traditional methodologies are not very effective. And I'm still working on the development part. And in fact, I'm very excited because at this point in my career, what I'm really setting up right now is a major project to test out how we could actually improve people's leadership by working the brain in a different way. This over the years has led to something called organizational neuroscience. And organizational neuroscience is because we came to a conclusion pretty quickly that studying your unit of analysis being a person, the leader, is not enough. A leader's got followers. A leader's got an organization or a team or something to work with. And then we found out that, hey, there are teams that work without leaders, but the team themselves have, some neuroscience component to it. So we actually defined the term organizational neuroscience I don't know, 12, 15 years ago. Nowadays, we refer to it as organizational neuroscience and biology because there are a lot of our colleagues that also do things like haptic assessments or chemical assessments, dopamine, and other factors that are not necessarily neuroscience, but are related to being hard science or life sciences associated with the social sciences. So my contributions is I'm an old timer. I started doing this when people thought I was nuts. And now there are over 500 people that are part of a special or interest group at the Academy of Management called Neuroscience and Biology, the newbie. And I'm very happy that it has grown and it has allowed me to travel the world basically and have connections around the world of people that have like interests. That said, close to 30 years into this, we are only touching the tip of the iceberg. And that's why I'm ecstatic to have an opportunity to talking to people that may be curious about this so that you know that this is very legitimate. Currently, I am sponsored, and I like to acknowledge my sponsorship. The U.S. military has been very, very important. They seem to be very intrigued with the human brains as it affects midshipmen and also how it affects people that are soldiers and people in society at large. And currently, my visit today here on campus is sponsored by the Army Research Institute. And I've got a project with them. I'll mention a couple of things so that you see right off the bat what organizational neuroscience really is. And it deals with radar memory. This is work that Bob Lord and Tiffany Keller and a couple of others like Paul Hangus at the University of Maryland have been involved with for a long time. And they're looking at memory. Well, memory and the brain have got a lot of stuff in common, right? So if you understand the different types of memories that a brain process can support, you can certainly contribute to that research stream. And I also am working with the Office of Naval Research. And this is the part where I'm really excited because we call it neuroadaptive optimization, i.e., training your brain to be better at doing something. And that's what they're heavily involved in. So going back to the first project, the one that I'm working on with Tiffany, Bob Lord, and a couple of others, memory matters. And in a nutshell, this is my contribution to something that they've been looking at for a long time now. So most leadership assessments, okay, so you're interested in assessing your supervisor. And we do this all the time. And most of us for generations have been using Likert scales and, you know, when A, B, C, D, E, and whatever, and my boss is like this and my boss is like that. Ultimately, when you're filling up one of these things, it's a memory activity. And you would assume that respondents can accurately retrieve the right information and answer the question appropriately. Now, memory science would suggest otherwise. And they have proven there's a lot of inaccuracies in how leadership assessments have been done, and very specifically within the military. So whoever gets the promotion to be a colonel or a lieutenant general or whatever, you'd like to have the right person get the promotion, right? So leadership ratings depend on how information is actually stored in the individual and how it's ultimately retrieved. And all of us have got different systems at work, and we look at the same information in different ways. So assessment quality is really affected by a couple of things. The system in the brains of individuals that are doing the assessment and how you construct your questions to retrieve that information. So the three types of memory that this research is mostly connected to are things like episodic, semantic, and procedural memories. And instead of going through all these definitions, I'm going to just put you in the frame of you are now answering a question about your boss. And I'm going to ask you essentially the same question three different ways. Okay? So we want to see if our boss is actually pretty good at supporting you as a worker bee, okay? So in many of our surveys that where you're assessing your boss, we would have a question like, oh, my boss is a supportive leader, A, B, C, D, E, okay? You put in the circle. Now, I could ask the question in a slightly different way. To what extent is providing positive support for you a accurate description of your boss's leadership style? Okay, so the first one is supportive, and you just answer. Next one is to what extent, and we go into more detail. Maybe I can ask you a question this way. Recall a specific instance during the past month when you needed help. How supportive was your boss during that event? Believe it or not, The same question asked 3 different ways is making a different part of the human brain work. And depending on which one of these brain profiles you're good at, you may or may not be very precise in your answer. So we're learning a few things. We're learning about the memory systems and how they are useful in getting an appropriate response to questions, in this case, assessing a leader. But number 2, we're learning about the science of constructing questionnaires. So you see that last part is really what the role of organizational neuroscience is. Because before that, you're basically looking at a paper product, okay, the survey, what worked, what didn't work. You might have measures on which one was more accurate or not accurate, but the organizational neuroscience part is actually giving you a real explanation of why this is, and actually giving you a recipe of how to improve as opposed to doing it in a less efficient way. So that's what organizational neuroscience is all about, as an example here with the Army Research Institute. Now, just for the fun of it, background, okay, facts about the human brain that everybody should be somewhat aware of, but it's surprising to take a look at one slide like this. For example, the brain, what's between your ears, if you took it out and you actually carried it in your hands, it's about 3 pounds of flesh, okay? So that's kind of interesting, but in that 3 pounds of flesh, it's about close to 75% water, pretty much like the human body. Basically, it feels like jello. The thing that's interesting is the cells that we've got in the brain, okay, which we call neurons, which are cells of the nervous system. There's 86 billion neurons. I used to say 100 billion, but the estimate has been revised downwards. Okay, a couple billion here, a couple billion there. Nowadays, the accepted number is about 86 billion neurons. What's interesting is every cell has the capacity to communicate with other cells. And that's called a synapse or a synaptic connection. And every cell has between 1,000 to 10,000 synaptic connections possible, okay, in each neuron. So if you did the math, there's 100 trillion plugs, okay, in your brain. That's kind of fascinating because that's a number that most of us, even if you're in quantitative whatever, it's a very big number, okay. Blood vessels. Note that if we, and these are very microscopic filaments, okay, there's 100,000 miles of blood vessels in the brain, in that same 3 pounds of flesh. And at any point in time, 20% of the blood in your body is in your brain, in that 3 pound thing. That's where the action is. 60% of your brain is fat. So when you take a person and say, oh, that's a fat person, well, maybe their brain is also very fat. It's kind of interesting to see as a number. It also consumes about 20% of the total energy in your body. And that, by the way, is enough in terms of wattage, and you'll see in my other slide, probably light a light bulb, if you could actually accumulate and aggregate all that energy. We also know, in an odd way, that the fastest speed recorded for communication between brain areas is about 268 miles per hour. So that's taking, fractions of seconds and figuring out how this part of the brain can communicate with that part of the brain because they need to talk to each other. And it's done in very, very, very quickly. So as I said, you can actually light a light bulb with the energy. Okay. Now, why is there energy? Well, they're chemical reactions. A synapse is, in fact, a chemical reaction. And every time there's a chemical reaction, a little bit of energy is extended, okay, from that chemical reaction. And there's also blood flow. And blood flow is also chemical energy. Okay, so all of this is going on in your brain. And this is what, over the last century or so, we've been trying to assess. Also interesting is the brain feels no pain. Okay, there are no pain receptors, so you will see sometimes on TV open brain surgery. And people are saying, can you feel this now? The answer is no, but if, let's say, you are affecting something, if you press something, maybe a person can't speak, but they won't feel the pain. that sort of stuff. It's fascinating, and I only know the tip of the iceberg. Now, rules that I have learned that I put right up front, especially when I deal with doctoral students, because they could be 2 or 3 years down the road studying very expensive, extensive stuff, I should say. And you go back to rule number 1. And rule number 1 is you don't control your brain. Your brain controls you. We believe as humans that somehow, okay, I as a researcher can do something to it. No, I can't do anything because the brain is actually in control of me. Okay, stop breathing. Tell your heart to stop beating right now. So one lesson you have to learn is you're not in control. In fact, the estimate is, and it's very difficult to figure out the exact number, between 90 and 95% of what your brain does, you're not even conscious of. So the unconscious brain is actually much more important than the conscious stuff. And that goes to rule number 1. You're not in control. The brain's in control. And if you know that, it can be helpful if you study the brain. The other thing that I learned very early on, some 25 years ago, when I actually ran a study, your brain burns energy even when you're doing nothing. Okay. Now, my big surprise is I was actually calculating, okay, the brain activity, also called power, in one of my very first experiments. And I remember calling my mentor, and I said, I completely screwed up. I'm so sorry. And he looked at me and he said, why are you apologizing? I said, well, I don't know. I probably inverted some matrix somewhere, but where I thought the numbers would go up, they're going down. And where the numbers are going down, they would go up. So I saw more activity in the brain when a person was sitting down doing nothing. And when I asked them to do some very big, math problem, numbers would go down. And the guy starts laughing at the other end. And you have to remember, it's 25 years ago. And I said, so I didn't screw up? He says, heck no. He says, Pierre, welcome to the club. You're one of the very first researchers that have identified the concept that in neuroscience, we call the default mode of the brain. Your brain, when you're asleep doing nothing, you think you're doing nothing, but it's busy like hell, okay, doing a lot of stuff. And when you're daydreaming, you think you're doing nothing, but your subconscious is going wild. And we know that because we can study what's happening to your brain when you're sleeping, when you're awake, when you're making a decision or whatever. And the default mode of the brain, which represents in terms of space, about 60% of your brain is active when you're doing absolutely nothing. Okay. So nowadays, a lot of research talk about the default mode. 25 years ago, it was a new phenomenon where people figured out that when you are doing an activity, it concentrates the activity in the parts of the brain that you need and shuts everything else down. And when you're not doing anything, the energy, okay, spreads to all parts of the brain and thus uses more energy. Now, your brain likes to conserve naturally. It's a lazy thing. So it prefers familiar ideas. Okay, repeating a habit, driving home tonight, okay, in your car. Do you ever remember, did I really make a right turn or left turn? Was the light red tonight? You don't remember. You're on autopilot when you do this because it's automatic. Okay, you've done it so often, and your brain puts it in autopilot. So your brain learns not for the fact of learning, but for the fact of, well, next time I face this situation, I don't have to work as hard. Okay? It does that all the time throughout your life. Okay, so I asked people, I said, do you remember when you learned how to ride a bike? Oh, not really. But take a look at a video of your kids learning how to ride a bike. Can you unlearn the concept of balance? No. Maybe the last time you rode a bike was 15 years ago. Somebody gives you a bike, you might not be great at it, but you understand the concept of balance. Before that, whoop, poof, you would fall down, but you learned balance, and then you got it. So the brain parked that knowledge somewhere inside your gray area, and you don't lose it. Okay, so that's what's amazing. And you don't have to concentrate, okay? Riding a bike, for most people, they don't have to concentrate. Oh, this is what I need to do to be balanced. No, it's now part of who you are. A lot of what I see in the literature is what I call neurobabble or neurojournalism. Okay, there's a whole generation that'll tell you, "Oh, you're so left brain." As a person that has done a lot of work with brains, that's BS. Okay, there is no such thing as left brain and right brain. However, for about 25 years, there are parts of the brain that are more into this part than that part. But what people are doing nowadays is they understand that there are networks in the brain responsible for different things. And these networks can be in the right hemisphere, the left hemisphere. They can switch around and stuff like that. But there's a lot of evidence that shows that there might be a dominance in one side for a specific activity, but there is, oh, you're so creative, you're so left brain. That part is neurojournalism. Okay, so you have to learn about that. And gut feeling. Okay, we all have gut feelings, you all have premonitions and whatever. That's actually your brain recognizing stuff before you consciously recognize it. And what I want to do at the very beginning here is to talk about consciousness for a second, okay? Because consciousness is what we as researchers like to do. We study things we can control, understand, and whatever. And a lot of the brain is beyond that tip of the iceberg. And I will demonstrate it to you very quickly, okay? And all of you are going to see this somewhat differently, okay? So for example, when you see something like this, many of you will look at this and see some movement, especially if you're at home and you're looking at your computer screen. Nothing is moving. Okay, it's called an illusion. This is also an illusion. Is the line straight? Most of you think it goes up a little bit. It's not. It's perfectly horizontal. Okay. The next one is actually used by a lot of psychologists. It's called a Stroop test. And that's how we see how your brain, different brain networks, interact with each other, don't like it, and want to learn how to best do this. So if we did it 100 times, you'd get better at it. But naturally speaking, I'm going to show you the next slide, and you're going to feel the fight, okay, in your brain, because there's one network trying to do one thing and another network trying to do other things, and they're all both on autopilot, and they're now going to hit each other. And this is called a Stroop test, okay? So the idea is you take a look at this and say the color. See, your brain is trying to do stuff, okay? And this is a subconscious. And psychologists will use tests like this to see, okay, your brain capacity and how it learns and whatever, and might do the same test tomorrow. And eventually you'll get to a point where you will learn to say and not, you know, a green, yellow, or a red, blue, or whatever. Now, the next one is the same thing, your brain at work. Okay, you're going to stare at this. For most of you, this will take a long time to figure out. What's the mistake? And you start taking a look at, you know, the numbers and whatever. And this is all over the internet, by the way. Okay. And if you take a look at it, and I'll tell you what it is, okay, there's two "the" in the question. Most of you didn't see it at all. Why? Because your brain, okay, puts it on autopilot, okay, it moves on. It says, oh, I know this. No, no. In fact, I'll show you how not stupid you are, because the next thing I'm going to show you, you shouldn't be able to do anything with this gobbledygook, okay? It looks like a freshman's essay before ChatGPT in your class, okay? And you will all be able to read this. Probably very well. And this is a classic study that as long as the first and last letters are in the right place, your brain, okay, just, it gives me a hell of a lot of respect, okay, for people that are editors. Because editing is concentration. You have to go beyond what your brain thinks it sees and self-corrects, okay, subconsciously, to actually see what's there and correct what's there. So that's why it's very difficult for you. You read one of your manuscripts, and maybe 2 years down the road, you see a typo, and you go, geez, I've read this thing 1,000 times. Yeah, but your brain was on autopilot. Okay, so that's why it's always good to read somebody else's paper. Now, I'm going to do a little bit of a priming exercise here, and at the risk of doing priming, I'm going to show you again, trying to show you something through an example. I'm going to show you a list of words. Okay, and the part that I'm not supposed to tell you, but today is a presentation about the brain. Think not just about the word, but put your brain to work about the meaning of what you're trying to be told here, maybe what the pattern is, and maybe take a look at what your feelings are about these words. Okay, because I'm talking about memory now. Okay, so I'm going to give you a list of words. Okay, so and it's appropriate. It's Friday morning, mid-morning. I think we should be okay with this. But the first word is bed. The next word is rest. The next word is wake, followed by tired, followed by dream, followed by snooze, followed by blanket. Okay, let's relax. Let's doze. How about pillow? Night. So here's the question. How do you feel? Relaxed, right? Okay. Did you recognize the meaning or a pattern? Yeah. Okay, good. So you see, even a word, your brain goes to work. Even if I didn't say do it, everything that happens in life, you connect your own personalized meaning to it. Okay. So this is about memory. Emotion strengthens memory formation. In fact, a few weeks ago, I've been using an example for years, and I actually checked out. Do you know that 9% of the population is actually scared of dogs? Okay, and I actually own a pretty large pit bull. And all my life, okay, and he's trained. He's probably the most gentle creature on the face of the earth. Okay, he's now 11 years old, and he's just a puppy, but people are scared of this thing like you wouldn't believe. But the thing is people that are really scared of dogs, it's because something early on in their life made them scared to the point where about 5% of the population have a phobia about dogs. So you see, your brain, it's not about this dog, it's about dog. Dog is a bad word. Dogs are dangerous. Dogs can hurt you. Why? Because something happened and your emotions and feelings are connected with that. So next time you see a dog, even if it's a poodle, oh, I don't like poodles, it's a dog. Okay. So especially if there was a very bad event at some point, amygdala is very deep in your brain, by the way. So when the amygdala gets involved, it's a visceral reaction. Okay. Now, your brain, and I kind of showed you, remembers meetings and patterns, okay, the feelings you had. But the thing to point, I'm trying to point out here is it will remember that better than the exact details. Okay. And if I do multitasking, it reduces memory encoding. And we see this every day, okay? And Omer will like this because he's the marketing guy in the room. And you know how much money we spend on marketing advertisements and whatever. And what's maddening is you may have seen the ad 1,000 times. And if somebody asks, okay, what company put the ad there, and you won't remember the company, but you can recite the words in the ad perfectly and how the ad makes you feel and whatever, but you don't know what product they were selling. And we also have learned to shut off our brains during, ads because it's not important stuff. And the more we see ads, the more our brain puts itself in autopilot. Oh, just disregard that. It's not part of the show that I'm watching, so I'm not even listening anymore. You might be listening and you might be going like this, but your brain is completely checked out. I've run experiments like this, so I know that as a fact. Okay. So that's called multitasking. And multitasking, okay, reduces your capacity to encode anything. The hippocampus, also pretty deep down in the brain, is associated with making something worthwhile remembering with more precision. So if something big happens in your life, you will encode it and you will learn the feelings, the meanings, and the patterns better than something that you didn't particularly care about. Okay? And the part that's interesting is memory is reconstructive. That means it's not a videotape. If you ask somebody to say something that happened to them 10 years ago, and it's the 100th time that they give you the story, the facts that they're going to give you are going to be slightly different. And that's why, you know, cops have got a very tough job, because they ask people, okay, what happened here? And they ask 100 different people. 100 different people saw different things. And you might ask the same person the next day and they saw something different. Sometimes it's more precise, sometimes less, because the brain has something also that's pretty fascinating. It will actually fill in the blanks when there's a blank. So if you can't recall something, your brain will actually do like ChatGPT. It's called an hallucination. Okay, it will just fill in with something that's pure BS. Okay, was the word sleep on the list? Okay, so you see, if I did not prime you, most of you would say, oh yeah, okay. But in reality, the word sleep was not there. Okay? We had doze and we had all types, but the word itself was not there. And that was meant to demonstrate that, you know, you may not remember the details, and frankly, who cares if sleep was not there? But you'll all remember that at the beginning, we were trying to get ourselves relaxed just by looking at words. All right, let's take a look next at some history. Okay, because, you know, the brain is something that fascinated me, not necessarily because I really want to know a lot about the brain, but anytime somebody talks about the brain, people listen. We're all fascinated about how we run our world, okay, how this thing, okay, controls us. This is not new. When you take a look at the history of who's looked at the brain, you can go to Hippocrates, okay, and there is some writings from that era before Christ, okay, of people taking a look at the brain. So there is some writings from Da Vinci on trying to understand the anatomy and what might be responsible for what. Okay, so we're going back a while now. Descartes, famous French mathematician, he understood that somehow brain, biology, and the mind, thinking, were separate. And still to this day, a lot of people say, can you read my mind? No. I can understand the biology of what's going on in your brain. That said, there are research projects that I'm aware of where they've made the link between a thought and the biological signature of the thought, and are trying to artificially create the thought through the biology. Okay, that's kind of on the scary side, but we've always known that I can say, oh, your frontal lobe is very busy, but I'm not sure what you're thinking about. So if we continue now we're in the 17th and 18th century. Okay, so now we're getting a little bit more serious. At that point, people are starting to look at the electrical property. Oh man, we can light a light bulb with the brain. Why is that? Okay, so that's where they start understanding that the chemical reactions created energy. Okay, this guy called Franz Gall, okay, one of these crazy German guys, he was very, very big for psychology. But in about 1790, well, 1800s, okay, between 1800 and 1850, one of the biggest pseudoscience, just like saying left brain, right brain, but in 1850, we were talking about something called phrenology. And phrenology, for us that are losing hair, okay, you're basically telling people, okay, exactly who you are, because phrenology was, we can understand human behavior by the shape of your skull. So by the indentations and whatever, they actually defined the seat of everything in your brain, because clearly your skull is protecting your brain, so it takes the shape of whatever's underneath it. And Bell and McGinty, okay, understood that there were parts of the brain that were more involved with motor sensory skills, and other stuff which were not motor sensory, which were either feelings or thoughts or whatever. So this we're starting talking about now a couple of 100 years ago. Now, what's neat is if you go to a good library, you can get a book, old book, probably yellow pages, on phrenology. And I did that. And it's fascinating how they wrote this as if it was real science. And it's more BS, but it's BS 1850 style. Now, from the 1850 to 1950 is where we really came to the modern neuroscience component. And here are three individuals that were involved with this. Number one, there was this guy called Florence, who really started studying the cytoarchitecture. Basically, what does a brain look like? And if you take a look, even today, you know, we got NFL football players, okay, that die young, and they donated their brain to science. And they bring it and they basically take a look at biologically what happened to their brain after they're dead. It's very difficult to study somebody's brain when they're alive, although that's what we're trying to do without causing any damage. But in 1850, we didn't do things like that. Now, the guy at the bottom, okay, Brodmann, he actually defined the brain by taking a look at the architecture of all the parts of the brain. And he defined it so well that today, 2026, researchers use Brodmann areas to define specific geographical positioning of something going on in a specific location in the brain. So that goes back to about 1909 when he first defined the human brain. The guy in the middle in 1846, a guy by the name of Phineas Gage, is often known as patient number one. And a lot of neuroscience deals with N of one. And you'll see my two examples here and you'll understand why. Phineas Gage, before we get into Gage, okay, this is actually, if you went to a medical book today, and you were looking at the brain, you would have a picture like this, and it's called Brodmann areas, okay, for our friend. And we know all the different folds in the brain and what their role is biologically. Okay, so if somebody, okay, gets a lot of problems in the back of the brain, okay, gets knocked out or whatever, they may have vision issues. There's a reason for that, because your vision functions are in the back of the brain. And your sensory motor skills, they've known for a long time, okay? It's kind of a slit right in the middle of your brain. Your emotional regulation is to the front behind your eyes, okay? The executive function is right up here. Most of language is actually on the left side here. You know, we know these things. And guys like me are trying to say, okay, how can that explain specific behaviors within organizations? Okay. But this is a nice pastoral picture of Cavendish, Vermont. And the one thing you should notice is these rails that are in the picture, they were actually installed there in about 1848. And we know that because this Phineas Gage guy that I mentioned, he was a construction foreman who actually was in charge of putting these rails there. So the rails that are there were probably installed by a team under the control of Phineas. And Phineas was responsible for running basically dynamite. So they go through mountain pass about 2 miles out of town, and they had to, you know, blow up the mountain so that the train doesn't have to go up and down the hill. It has to go straight. And his job was to put dynamite and taking a tapping iron and putting it in there, very solid, whatever, and then light the fuse and it goes boom, the rock goes down, we clean it up and we build the rail track. Well, the poor guy, one morning it went boom. And this steel rod went right through his skull. Okay. Believe it or not, the guy walked to the Cavendish doctor's office, okay, as medieval as it was in 1850, and he says, "Doc, I got a problem." Okay, so you know you're dealing with unique situations where somebody's brain and part of his face were blown off, and the guy is making a joke about it on, you know, the table there, saying, "Doc, what can you do for me?" What's amazing is this guy lived 12 years beyond the accident. But everybody says he was a different person. Before that, he was, you know, in the gym, very jovial, very manly, very, you know, outgoing. okay, strong. He was a supervisor of men and whatever. And then he became pretty much of a recluse. So a lot of his behaviors changed, but he still was well enough to actually run a stagecoach in Chile on a daily basis for 10 of the or so years afterwards. And he, in fact, died 12 years later in San Francisco of epileptic seizures. And this guy, 20% of his brain was blown away. So people like this were very responsible for us to learn about, number one, how is it that this person, they survived? A lot of people did not believe that you could survive a direct attack on the brain, and he's the exception. To the point where today, if you go to Boston and you go to the medical school, okay, they've got a library with the history of medicine. This is this guy's actual skull. And the tamping iron is in the display case there. Okay, so you can see that. You just go, okay. Now, we don't have to go that far back. Okay. I'm always reminded of where I got my PhD. My PhD is from the University of Arizona. And this summer, okay, Savannah Guthrie, okay, brought it back home because that was the neighborhood I used to live in. And that neighborhood is very peaceful and nice and gorgeous 99.9% of the time. But a little while ago, at my local Safeway, the representative of the time by the name of Kathy Gifford got shot. And we all know Kathy Gifford, okay? She's now a gun activist. Her husband is senator of the state of Arizona, and so on and so forth. She's alive. And not only is she alive, she's doing pretty well, considering a bullet went right through one of her lobes, not just lobes, but hemisphere, from the back to the front. She does see. The bullet went right out, okay, from the front, right above the eye, but definitely has speech impairments, has physical impairments, has to walk with a cane. But compared to a week after the accident, or not accident, the incident, okay, and today, it's amazing because what it shows is the incredible power of the brain to learn a different set of pathways to achieve something. So it's difficult. Now, it's easy when you're a baby. I remember when my kid was learning French and living in North Carolina, people would say, it's amazing how kids can learn a foreign language. Well, some of you have probably the same experience, but for them, it's not foreign. Now, trying to learn a foreign language when you're 50 years old, that's tough because you've lost the capacity to do that largely, but not entirely. But your brain is capable of learning new things. And that's part of what I'm trying to do. Now, how do I study the brain today? Well, as I said, we're not studying dead people. Studying ends of one is not very good in organizational settings. So I can talk to Gabby Gifford all day, and I'll learn a lot about your brain, but I won't learn about how does the brain help organizations. Okay? A lot of neuroscience research is based on very small ends because they're looking at exceptions and weirdness as opposed to, organizational phenomenon. And that's a point. Now, for 20 years, I've been doing a non-fight of this issue. People say, oh, you should be using fMRI. Okay, there is a bias for fMRI research. And take a look at this. This is what an fMRI is. Okay, it's a $1.5 to $2 million machine, typically at a research hospital from a big research university. And take a look behind the shoulders of the lady. Okay, these are all different appendages to make sure your head won't move. And this person, okay, is strapped down and whatever. And then they roll you into this magnet. And it's a loud thing, okay, to get in there. And what it does is it basically checks the blood flow, okay? Because if there is fresh oxygenated blood in your brain, it has a different magnetic signature than older blood. So when your brain is being used in this part as opposed to that part, the blood freshly oxygenated, okay, goes and rushes to where it's needed. So fMRI studies take a look at blood flow in the brain in real time. But my question to you is, if you're working with the leader of a CEO or CEO of a large company, okay, a leader, are you going to make an appointment with them to go to the local hospital to get strapped down to learn how to improve their leadership skills? No, it's the wrong technology. It's a great technology for neuroscience. It's the wrong technology for organizational neuroscience. Now, the neuroscientists are going to say, well, your machines are not as precise. And whatever. We can get into that debate. That might take 2 hours just there. But do you know that any single thing that your brain concentrates on will take a minimum of 5 million neurons working together to get accomplished? So you don't need to go to the neuron specific. Now, if you're doing epileptic seizures, maybe. But if you're dealing with organizational phenomena, behaviors, and stuff like that, you don't need that level of precision. But what you might need is a person that's more than happy to sit down at a table and have something on their head. In fact, the people at the bottom was a classic research study that we did in Spain, where we actually studied the brains of several people working at the same time on an issue. Try doing that with an fMRI machine, okay? Now, what's interesting in this picture is you see several generations of what we put on people's heads. But what's important there is, hey, people are doing what they're doing. They may be in an office environment. They may be in a group. They may be, look, the military uses these things with helo pilots. And they test on an ongoing basis how busy your brain is. And depending on how busy your brain is, it offers you different telemetry in the helicopter. So if you're in combat, it gives you just the basics so that you can keep flying. And if you're, I guess, flying over the Potomac and you're trying to make sure you don't hit planes, you get a full array of everything, including what's the temperature outside and the wind speed and the whole 9 yards. And it does that automatically by figuring out how busy your brain is at that point in time. So in the field, it's a great phenomenon. Now, the biggest problem that I've seen, and you can see the bottom at the right, that's my one of my undergraduate students, Travis, about 15 years ago, and that's the chaplain of the U.S. Army, and a cap very similar to the one that I've got here. And what this thing has is a bunch of electrodes on it, and these are very sensitive. They can capture each about a millionth of a volt. So it captures everything. And the thing is when we used to do this, you can see Travis, he's working. He's actually squirting gel on this person's scalp to make sure that there's a connection. So my problem for the first 10 years of my career doing this had nothing to do with understanding, the data. It's trying to get a subject that's stupid enough to sit in this seat and having a bad hair day. Because you're going to have a bad hair day. You're going to walk off, we're going to take off this thing, and you've got a head full of gel. So you need a shower. Okay, so most people are not very interested in that. So over the years, we've done some development, and we've been involved with this from a technical perspective. The picture on the left, this young lady is wearing what they call a CGX-20, which is essentially the same thing as this, except for two things. This has a nice big wire that you connect to something called an amplifier. So you need to know what it senses and then make the numbers bigger so as humans we can understand what the numbers mean. Okay. So this particular machine that these ladies have, okay, the one on the left, the amplifier is not a separate machine. It's actually connected to the device itself. And you don't put any gel in the person's hair. Now, that particular cap, and I didn't bring it today, but it's about $30,000 for a cap. Spring-loaded, actually quite comfortable to put. We did a year's worth of research with caps like this, and we found out that the impedance, and here's another word, impedance is how good is your connection between your metal electrode and the skull. Because if the connection is not perfect, you get a lot of noise because it's so sensitive. And what we found out is this is great, expensive new technology, and it's good to collect power information, the amount of activity, but it's not good enough to collect data about the relationships between different parts of the brain. So the old-fashioned put gel in your hair gave me higher quality data at $750 a cap than the $30,000 cap. So for 15 years, I was bitching, okay, to the people in this technology saying, you need to find something that will work for people other than people stuck in the hospital bed. So they came up with, and a friend of mine in Munich, Germany, has developed what we call a semi-dry electrode. And instead of putting conductive gel in your hair, we basically put saline solution. Same thing you stick in your eyes if you've got, contacts. So little sponges, and you take it off, and you take a Kleenex, you wipe, and you're ready to go. Fantastic. So this is newer technology. So the person on the right is using a saline solution, 19-channel thing. Now, all of this, and just to recall, we're trying to sense the electricity or the energy created by the chemical reactions of neurons talking to each other. And there's 100 trillion connectors out there, and they're not all active at the same time. Okay, that's what we're trying to do. Now, note that all of these caps, regardless of if it's 30 years old and one of these, or if it's one of these sophisticated ones, have a very specific pattern to them. Okay, because how can I compare my data to the data of somebody else doing the same thing somewhere around the world? We have to have an agreement. Okay, and the agreement is what they call the International 1020 approach. So this is state of the art, sort of where the electrodes need to be, so that when I say, in comparison to this electrode, this is where that electricity came from, we all agree on the source of it. So this is called the 1020, and it's all based on the fact that some of these numbers at the base are 10% from either the Indian or Nasian, okay, the crook in your nose, or 20% in between them, one way or the other. So you cover the whole brain, and then you write software that knows this. Every electrode on here can take data from about 6 centimeters below it. So it covers the whole cortex and part of the more, what we call amphibian part of the brain, which is this amygdala and the hippocampus and parts like that. And this is what it produces. Okay. Now, there are some people on the face of the earth that claim that they can read this. They're called neurologists. And I'm fascinated. Okay. I can't read this. Okay. I need software to tell me what it means. But I can tell you that there are patterns, and I can tell you if it's clean or not. This is a pretty clean, result of what's going on in every one of the 19 electrodes. Now, at the bottom, you see that there are lines that are basically doing nothing. They're called aux1, aux2, aux3. You can put, for example, an EKG, okay, if you wanted to know if somebody's heart is doing this. Or some people take a look at eye movement or pupil movement and breathing. You might have a chemical analysis of what's going on. So that's where the biology component. Most of these softwares will allow you to have up to 4 channels, okay, of data. The thing is for me, this is show. I look at this only when I am taking somebody's EEG, because if I see this thing going like this, something's wrong with one of my electrodes. Maybe it's going like this on only one electrode, so then you have to fix the electrode. But don't ask me to read this. I'm sorry, no education on this whatsoever. So I can recognize patterns, but that's about all I can recognize after doing this for 25 years. What I can tell you, though, is there are frequency ranges. Okay? You've got, at any point in time, in your brain somewhere, very, very slow electrical patterns, delta, prevalent when you're sleeping, especially when you're sleeping pretty hard and you're not in REM sleep. If you're in REM sleep and you're dreaming, those of you that talk in your sleep and whatever, you're in theta mode. And theta is fantastic in terms of codifying your memory. Your brain is actually going through all the, what's happening to you today, and it's actually working with your hippocampus and trying to, put short-term memory into long-term memory so that you can recognize this. Most of our daily life, we are in alpha and beta. Alpha is called being in the zone if you're concentrating, okay? If you're doing something and you're comfortable and you can work, and sometimes you look at the watch and you say, wow, I've been at this for 2 hours and time just went like that. You were probably in alpha. Beta is you're really concentrating on something, okay? And gamma is super high-end concentration where you're using many networks in your brain at the same time, or that 268 mile per hour that I talked about, that's in gamma range where some part of your brain is talking to another part of the brain. So that's done in gamma. Now, when I tell you that I can't read this, okay, can't read this, you'll understand why I put this next analogy in there. If somebody says, what's happening currently in the face of the earth? Okay, you go, wow, there's a lot of stuff. Half the earth is sleeping because it's the middle of the night. Half the earth is awake. Okay, delta, beta. Okay, let's take a look at, I don't know, Manhattan right now. Okay, so you take a look at one specific spot on the face of the earth. On the brain, we would refer to this as a voxel. Now, if you take a specific coordinate within Manhattan, there are some people that are probably still asleep or dozing off in their apartment. Some people stuck in traffic looking at their cell phone. Other people that are jogging in Central Park, okay? So you've got all of these activities going on at exactly the same time, at the same spot within Earth, and the whole Earth, okay, is doing its own thing. But you can still see patterns. You can still understand, okay? Is it busier right now? You can actually figure out if you had some plane at 50,000 feet with a very sensitive microphone, oh, parts of Europe are in the siesta, probably mid-afternoon, and then it picks up at rush hour, okay? This is why I use this analogy. Looking at the brain and trying to come to grips with the fact that all of these different speeds of brain and power and activity and amplitude and whatever are going all around the brain all at the same time, and you have to disassociate this to understand what's going on. Now, it sounds complicated, and the neuroscience people would like to make it very complicated, but let me kind of go back to the very beginning. I am not a neuroscientist, okay? I graduated from the University of Arizona as a systems engineer, okay? So I know engineering, I know systems, computers. I always loved data. I was also a person that got a degree in information systems. But I found out I didn't like information systems. What I liked was, what type of systems do humans create to make themselves and their organizations more efficient and effective? So I'm never the guy in line at the Apple store to get the new phone at midnight. It's not me. But I'm very, very interested with what technologies can we use to make me better at what I do and be more competitive than the competition, okay? So you look at this, and to me, this is a godsend. It's called a quantitative EEG system. I don't care about the squiggles. I care about specific spots in the brain because of our friend Brodmann that said, hey, you should study here or you should study there. Okay, that's great. But take a look at the extreme right. See these numbers? Those are numbers. They're not a squiggle on the line. They're not a red picture of a human brain. They're a number. And that's about as core as I can tell you about what today's lecture is all about, is if you can understand that ultimately a brain map is just a set of numbers that you can put in your SPSS or SAS, half the game is over. So what I learned is if I know where to look and what to look for, I can get numbers. This software will create numbers about everything going on in your brain 256 times per second. So it's called big data for those of you in IS. But nowadays with AI, you can program an AI to take a look at this and it'll tell you immediately what's hot, what's not hot, where is the activity, why is it there, and so on and so forth. And the color there is, telling me something about, well, that's where most of the activity is going on right now in response to this. Okay. And other examples, same software, you can actually take a look at the brain and dissect it virtually and pull up again to the right a bunch of numbers if you're very interested in that area and what that relationship is. Now, I mentioned a while ago that one of the CAPs was not very good at doing relational data. It was good at doing power. Power is this. Power is, oh, this is where the brain activity is right now. This one is different. All the little white dots that you see are the center point of different networks in the brain. Now, the network that it's highlighting right now, if you take a look at the right, there's a check mark next to the default mode network. We've learned about that already. When you're at rest, 60% of your brain, and look at all the little white dots on both sides. So most of your brain is an active participant in the default mode. That means it's very active when you do nothing at all. Okay, the default mode. And I'm now concentrating on high levels of beta, which is a subset of the beta range. That means a person is awake, active, taking a look, the eyes are open, and so on and so forth. And at the top, on where it says menu, it clicks under coherence. Coherence is the neuroscientific word for what us in the real world would call connectivity. It's basically assessing, is this doing largely the same work as that place? So where you see kind of a hint of red and blue, and some is a little darker red, some might be darker, lighter blue, it's actually telling me where these different parts of these networks are doing largely the same thing at the same time. Are they working together? Connectivity. And what we have learned over the last decade is power is interesting to look at, but it's not the right thing to look at if you really want to understand the human brain. You have to understand connectivity among and within networks. So it's all about network neuroscience nowadays. Okay. So this software can be put into connectivity mode, coherence, and give you a variety of numbers pertaining to how similar or dissimilar two or more regions of the brain are working together or not. So I can actually come up with a number of how coherent is the default mode network. How coherent is the salience network? How coherent is the working memory network? How coherent is the pain network? And so on and so forth. This is called diffusion tensor imaging. So these are all white filaments that are throughout our brain, and they're basically the interstate highway where the 268 mile per hour occurs. And this is an analysis of how healthy or how busy these different highways are right now. Okay. And again, the important part is the picture looks great, but what's interesting to a guy like me is the fact that I can generate numbers and I can tell you which one's better, which one's not better, and actually stick an actual number associated to the phenomenon, and thus can be plugged into your SAS or your SPSS as a relationship to something else. And as I said, we can do this in groups also and see a whole bunch of people at the same time. And the software, which you can see here, actually times everything. So you can actually take a look at brains working either in tandem or whatever. And what we found with this study is fascinating. One of the papers we wrote was about the emergence of leadership in a team. And your brain knows very quickly in the first 10 or 15 minutes of a meeting, and these people did not know each other at the beginning, okay, but they worked with each other for a couple of hours. It took about 15 minutes for brains to figure out who's the idiot in the group. And it's funny, okay? This person would talk and all the brains would shut down and people would, you've got the video, it would go. And they were completely disengage completely from what this person was saying. And then the other person that was identified as being, okay, this person's coherent, doing some intelligent stuff, they would turn around and the brain would just react immediately. So your brain knows stuff even before you consciously know the stuff. And it's fascinating. So imagine, think of research projects you could do with that. Yep. Absolutely. Oh yeah, take a look at this picture. We've got numbers in front of people, so it's connected to the EEG. We also had a recording, and we also had a couple of other stuff. All of their conversations actually generated probably 2 or 3 doctoral degrees of people that did analysis of the words they use, because we wanted to know things like, what word made you intelligent so that people actually listen to you? In fact, there's one paper we started. Remember in the 1970s, there was this saying, those of you that were around, when E.F. Hutton talks, people listen. Okay, this was a branding of an investment firm in New York. And we started the talk, our paper, by saying, when E.F. Hutton talks, people listen. But when Bob talks, nobody does. Okay. And it's not listening because people were going like this and doing the right social response, but the brain was completely checked out, and vice versa, which was fascinating. By the way, this cap is not a full 19 electrodes. This only has about 10. It's a company in San Diego. In the United States, it's amazing. Most of this technology comes out of San Diego. There's 5 or 6 major firms. And this is Advanced Brain Imaging. Very good company out of the San Diego region. We started doing stuff with caps like this, where we put a bunch of what we call, goop or glopper in front of people and say, hey, you see all these things? Tell me a story about the development of your life using these things in front of you. So we took a guy like a financial industry executive, and we wanted to see novelty. So here's a bunch of stuff you've never seen. We give them 5 minutes, take a look at all of these weird things we put in front of us. Okay, use these things to tell me a story about your life. What? I'm a financial expert. Yeah, we give you a novel situation, and we were trying to see how quickly people would resolve. We'd also have things like this, where we actually have a very interesting paper on vision. Okay, so leadership vision. And this person was very inspirational. And we had a question at some point asking about why would somebody work for your company, and where do you see your company 5 years from now? And let's see if I can get her to speak right now. Maybe I can.
Our work is inspiring to start with. Cleaning up the planet Earth is one of the greatest visions we have on this earth, and people love doing it. So it's easy for me. Everybody wants to come to work at our company. So the vision is simple. We only have one planet. Let's take care of it. Let's clean it up. There's nothing greater to observe is when you've taken a highly contaminated site and now it's usable for animals, plants, humans. Now the water's drinkable. I think it's a great feeling, and the team all enjoys observing that transformation.
So when you have a subject like this, okay, where we ask a question and everybody on the research team goes, whoop, okay, they wake up because here's somebody that's actually not giving us the regular good old BS, somebody that says something coherent, okay. We circle this, and we've got doctoral students, okay, taking a look at the brain profile of people like this. And we also need people that are completely befuddled by the word vision, okay. We've had people that are strong leaders from a position standpoint that couldn't say anything about the vision of their company if their life depended on it. So we've done a lot of that work. Now, the thing that's interesting to know is when we ask a question like this, and this person is using a dry electrode system, we always gave them between 10 and 20 seconds to put their thoughts together. So typically, eyes closed. That's why it looks like this person got their eyes closed, because they're there and they're concentrating on what they're going to say. That's the cleanest form of data that you can have, because if you recall, I said every electrode is a millionth of a volt. So just moving your tongue in your mouth will be captured by this technology. Okay, so you have to minimize movement while you are trying to clean up your data as much as possible. So we gave them, and we've been very successful at getting executives to basically concentrate, and we say, okay, you may now provide your answer. We're still recording, but there's a lot more dirt in the data when you're moving and doing all types of stuff. Yet, we record the answer because the answer has been thought of, and there's a high correlation between what they were thinking and what actually comes out of their mouth a few seconds later. Okay, so we've done things like this. Now, note, everything I've talked about right now is not really where most of my research has been. It's what people understand. And what people understand is what I call model 1 of neuroscience, which is what we call the functional neuroscience. Essentially, we strap you up, we do something to you, and we want to see how you react. Now, there's 100 trillion synaptic possibilities. So to me, and it's been proven to me by my mentors, that it's much more difficult to do a clean, functional, on-task neuroscience study. Because if you have somebody in the fMRI machine and the lights flicker for one subject, but they didn't flicker at the exact same moment for the other subject, you've introduced something in your experiment that's different, and something in the 100 trillion synapses, okay, caught it. So that's just to say that I've been much more successful in my research to take a look at model 2, which is basically your brain at rest when you're doing nothing at all. So I am not looking at how you react to something, but I'm looking at your basic capacity. What's your structure of this network? What's your structure of that network? What's your structure of this network? With the understanding that if you've got a good structure, when you need it, it'll be there for you. But if your structure is poor when it's doing nothing at all, it's not going to be there when you need it. Okay, so it's kind of like if you check out NASCAR vehicles, and one car has got 700 horsepower and the other car's got 400. Well, the 700 horsepower is a faster car. Okay, so my research has been largely, although every experiment we do, for the fun of it, we do a functional measurement, everything that's gotten published, okay, with the cleanest data has really been intrinsic at-rest behavior and basically capacities of different functions. And that's where I'm going to go with this. Now, the next big deal, okay, in my presentation today, I'm going back some 14, 15 years when we did this research that became the paper of the year in Leadership Quarterly in 2012. This was fascinating to me. We had an opportunity of working with the Army research the first time, not the research I'm talking about now. And we basically had 200 people of the US Army go through an EEG protocol. And we did all types of other stuff with them. But the basic study was, who are you? How good do you rate on leadership? And we have your brain map. Thank you. Next. Okay, so we did 200. And with my neuroscience mentor in Florida, he showed me how to basically come up with the brain profile of a phenomenon. And the phenomenon could be anything. In our case, we decided to use something called the multi-factor leadership questionnaire, MLQ, that had been used for 30 years to assess leadership. Okay, so we scored everybody on the MLQ, and we had their basic brain profile. Okay, and what we did, for those of you that are statistically minded, was something very interesting. It's based on basic discriminant function. So on one side, we took all the neuroscience variables that we had on that subject and put in one pile, and then we had the MLQ on the other pile. And we said, do a discriminant function. Which one of these variables could we put together that could predict this number? Never been done before. Okay. And we thought we'd get garbage. So in a research methodology, we took the first 100 and we basically said, we're going to train our process. So we used the first 100 to come up with the discriminant function. And all the discriminant function says, which variable should you keep? And what weight should you put on each variable? That's all what a discriminant function. And some of you do this every day with all types of stuff. We just decide to use brain data. Okay. And this is what this next slide is going to show. At first, concentrate only on what's black. Every time I put this slide, I look at the last line, I look at a typo. If you look at the journal, the typo is there, okay? But what's in black here is, these were the QEEG variables available to us in 2011 and 10 when we did this research. Okay, so we had all the variables, okay, of a person's brain. It's a grand total of 10,393 possible variables. Okay, and we ran a two-step process to figure out which one of the variables we should keep using a discriminant function approach. And it spit out a grand total of 58 variables. So if I took somebody's brain map and I took out these 58 variables and I associated with those 58 variables the weight that the discriminant function gave me, I could predict what the score would be on your MLQ. Now I'm looking at this and I'm very skeptical. I'm going, oh, this is bullshit. This will never happen. Okay, this just, and believe it or not, we could, in fact, separate people that are very good on MLQ, people that are not so good on MLQ. And when we took a look at the brain profile, it also separated in two groups. So a person that was Midland did not fall into two groups. They were Midland. So we didn't know exactly what their score was, but it told us that our discriminant function actually created a linear approach. You're either good, bad, or you're somewhere in the middle. And when we did the full analysis, we came up with an incredibly good score for something that was so rough. Okay, this has an R of 63, which basically means it's a very good predictor. And in fact, I can tell you that we refined it a little bit, that I could take somebody's brain map and say, well, on the MLQ, your overall leadership score will be between 4.1 and 4.3. And we were on it most of the time. Isn't it? Okay. Now, let me go back. This is good. Okay. If you ask Bruce Avolio, who actually is a co-author on MLQ, he's going to say, no, I can do early career, mid-career, late career, and they get about the same score. But if you ask me, I would say, yeah, you can change that. And I'm actually very different than most people. When they say, is leader, are you born? Are you, or can you actually develop your leadership? The answer is yes. Okay, you can change your brain. It takes time and effort, but you can. Now, take a look at the same chart, but now in red. Let me tell you why I'm excited and why I think the Navy is going to get a good deal out of this. Since we did this in circa 2010, 11, 12, the machines that we use are the same as today. What has changed is the model behind them and the software. I had 19 channels, and I split the brain in 2,394 little voxels, little boxes of your brain, and I could be as precise as that. Nowadays, the model has changed. So the same brain produces 12,470 voxels, a lot more precise. And instead of having 10,393 variables, my choice is over 500,000 variables. So I don't know if we're going to have 58 or 580, but it's now a level of complexity that I've never dealt with before. Okay, and because we have AI and stuff like that, we can approach the subject theoretically the same way, but get 10 and 20 times and 100 times more precise, an order of magnitude more precise. And it's going to be fascinating. The other thing that allows me to change this is because we've got so many variables, instead of just looking at the whole brain as being one thing, because the 58, variable, the biggest disappointment for me is there was no rhyme or reason, especially on the power, the first top part, which are individual brain locations. The bottom part is the network analysis, where everything is nowadays, and there's a reason for that, is when I took a look at where the variables were selected, it made no sense. When we took a look at the variables in the network, at least you could recognize certain network. Okay, this are in that network. This are in that network. The other one is like, oh, the delta in this part, at that second versus the high beta in this part, and you go, there's no link. It's just random. It's just mathematics. Okay, the network part is much more interesting. So instead of taking a look at the whole brain now, is network neuroscience has moved so much that we know at least of 12 fundamental networks in the brain. And each one of those networks is going to give me at least 5,000 different variables to look at. So we're going to do a discriminant function of each network in the brain, as opposed to considering the whole brain mass at once. So we'll be able to say, oh, when you're dealing and you want somebody that's very good at X, they need to optimize their semantic network, or they need to optimize their pain network, or whatever. So it changes the ball game, but it's the same thing. In terms of, are you high or low in leadership? If you take a look at our R there, okay, it was 0.85. We had a greater than 95% classification accuracy. So just by looking at somebody's brain map, we could say, okay, this person's a leader, this person's not a leader. So that was kind of exciting. Now, over the years, using intrinsic brain, we can identify a person that's calm under pressure. So the person that you want to be the captain of the team in a high pressure situation, we can figure that out. Visionary leadership, we are very good at that. We know a lot about transformational leadership, and what we do know is not all factors of transformational are as good as they pretended to be some 20 years ago, but there are certain subsets of transformational leadership which are very, very well understood. Leader self-complexity. Army is very interested in self-complexity. We know a lot about ethical leadership. Okay, there is actually a couple of networks in the brain that are associated with people that are ethical, understand ethics, which is very important in the U.S. Army. Social astuteness. Okay, people that are very good at understanding people. It's very clear. And we had a paper that was somewhat controversial, but the science wasn't controversial, is how to actually paint it. We had a paper on abusive supervision. So it's very clear where the seat of abusive supervision, what networks are involved. And it's very deep in the brain. So it's not something that you can just take a weekend seminar and stop being abusive next week. So if you have an abusive supervisor in your life, it's something that's very, it was learned early on in their life, probably in the first 5 years, probably when mother could not correct the kid and they were always getting their own way. Okay, another way of saying that you can't change Donald J. Trump. He is who he is, and no amount of neurofeedback is going to change this guy. It's deeply rooted. Okay, and note that why it was controversial is this paper came out while Trump was in his first, foray in the White House, and he was attacking his attorney general on a daily basis. Okay, he was basically the poster child for an abusive supervisor. Okay, and the paper comes out in the Academy of Management. And, it was kind of an interesting time in my life. Anyways, in 2026, hopefully now you can understand a little bit better when I say, look, modern QEEG software, okay, can study 88 Brodmann areas and beyond. You now know who Brodmann is. I now can look at the brain by splitting it into 12,470 morsels and understand all the electrical profile of what's going on in each profile. And I can build the blocks together and say, okay, these voxels are part of this network and these voxels are part of that network. And I can now look at it and get network level insights. Discriminant functions, the same stuff as you learned in Stats 101, is what I use. And it's based on strongest correlates of those 500 plus thousand variables, where it was about 10,300 before. And instead of examining the whole brain and trying to figure out, okay, what's the seed of this, what's the seed of that, I've learned that that's not the way to do it right now. We basically study 12 networks, and I'll show you in the next slide which networks I'm looking at. And the variables I'm looking at is more not activity level, not power. The basic variables for a specific location is power and amplitude. Basically, what's the speed, what's the profile of the electricity. What I'm more interested in is a variable called coherence, which is, neuroscience speak for connectivity between spots in the brain, and something called phase. Because phase is very important. The whole brain is about recruiting, recruiting neurons to help you with a problem. Okay. So when in an fMRI machine, it says, oh, the blood goes from here to here. And when the EEG says, well, the electricity goes from here to here, they're basically saying that some part of the brain needed another part to, take care of an issue at hand. And because of that, it recruited neurons from that other network. Okay, just like when you were dealing with your green yellow. Okay, you looked at it, took a few seconds to figure out what the problem was, and then you started figuring it out and you were slow at it. And then if you redid it, you would improve the connectivity between those two networks. And that's called phase. Your phase, or the recruiting of neurons someplace else in the brain to help you solve the problem, okay, it could be slow at the beginning and it will speed up. And that's why we take a look at phase measures. Okay, and also once you're done with the use, you want to release those neurons. And the brain loves releasing neurons because, as you know, the brain is lazy, tries to automate stuff, tries not to concentrate and work too hard on anything. So these are the 12 networks we look at. I mentioned salience very quickly. Executive function is at the front of the brain and deals with basically controlling what's important right now, what's not, what should be put on ice, what's immediate. Default mode we've talked about. Attention network. There are two attention networks. There's a dorsal network and a ventral network, and they're responsible for slightly different things. We've talked about memory today a little bit. We didn't talk about mirror neuron systems, which is how, as a baby, we learn what's acceptable behavior. And people that have got good mirror neuron systems are actually quite sociable because they understand very quickly, oh, I can't do this. One of the research projects in my future will be dealing with global mindset at Arizona State. I've been working with global mindset all my life. We are fascinated with people that are very good at dealing with people that are not like them, that come from not only different language, but different cultures. And we suspect that people that are good at just being a global citizen have got a very, very good mirror neuron system. Mood regulation, the anxiety network. There's also a pain network. And believe it or not, pain network is not just when something hurts. It's how you manage things that, another word is, are you resilient? Okay, so pain could not necessarily be physical pain, it could be psychological pain also. Language networks, and also addiction network. An addiction network is also a positive thing. When you are really committed or motivated to do something, it does a chemical reaction in your body that you like. Some people, have to get that chemical reaction artificially through drugs or alcohol. But a lot of people, okay, get all their endorphins by doing something that they thoroughly enjoy. Okay, ask people that are runners, why do you do it? Feel great. Okay, so that's their alcohol hit. Okay, so that's why it's still called an addiction network. Yeah. I'll send you my paper and you can read about it. I'm not sure it's a network per se. It's part. There's a couple of parts of networks. Many of these things, by the way, dealing with leadership, you will see that there's a contribution of several networks. It's never just one network. What I've learned when I first started doing this is I want to know where in the brain is the location for leadership. 30 years later, I look at my old self and I go, how stupid was I? Okay. People that have got a very homeostatic, well-balanced brain across networks are probably the people that understand right from wrong the best. But I'll send you the paper. It's fascinating stuff. So nowadays, okay, instead of just looking at the brain as a whole, we are going to be looking at all these networks independently. And what I've got here as you know, a speedometer is if you're in the green, it means your brain is actually in a good location for that network. If you're in the red, you need work. So you see, the one thing that I alluded to earlier on, which is mostly exciting to me, is we are at the cusp of doing part 2 of the definition of leadership neuroscience, which is development. Okay? We know how to change the brain. For the last 30 years, people have been correcting kids with ADD. So there are profiles and softwares and whatever. It's just that we have to apply it to a different problem. How to become more attentive, how to reduce stress, how to be a better decision maker under stress. Okay, a variety of things like that can be identified very specifically as what capacities have you got to do a good job with this? And if you don't, what do we need to do to improve your brain to support that capacity? Okay. And that is something called neurofeedback. Okay. And from my perspective, I'm unusual in that I'm interested in EEG biofeedback for non-clinical purposes. So it's based on the electrical brain activity and trying to figure out how do we optimize these networks that we've defined. It's directly training your brain. But don't forget, you're not in charge, your brain is. So let me go straight to my next slide. I'm going to tell you how weird it is to do neurofeedback because you're not in control. Okay. Take a look at the bigger picture on the right side. Okay, this young lady, okay, is looking at a video. Well, when I got introduced to neurofeedback the first time, okay, completely skeptical, okay. The guy puts me in a room very similar to this, large TV set in front of me, and in front of me, okay, is a dune buggy, and it says start. And I'm expecting any second that he's going to give me the joystick, and I'm going to be driving the dune buggy, right? So he's putting this cap on me and whatever. He goes to the computer and he does something, and he says, okay, Pierre, I'll see you in about 15 minutes. I go, Doc, wait a sec. Where's my joystick? He chuckles and walks away. So you're in this room, sitting down. You know that you're supposed to do something, and in front of you is a TV set with a dune buggy and a start flag, and you're supposed to drive the dune buggy to the finish. Okay, so you make these assumptions, and you're staring at this, and the first couple of minutes you really feel like an idiot, right? What am I doing here? So you start staring at the dune buggy, and you start doing all types of, you know, faces and whatever, going back and forth like every normal human being would do in that situation. And at some point, something jerks on the car. And you stare at it again, and it jerks again. Believe it or not, within a couple of minutes, you will learn what your brain needs to do to make the car turn right, turn left, go frontward, or go backward. And when you're all mixed up, it just stops running. So what this guy programs in the computer is what I want the brain to be doing and give a reward to the person when they do the right thing. So all you tell the brain is, I want you to win the race. And your brain wants to do good. Our brain wants to do good things. Okay. So by just telling the brain, you now have a goal. You don't know what the hell, because you're not in control, your brain is. But your brain is learning what the algorithm is at warp speed. And within 5 minutes, here I am driving a dune buggy, okay, down the dunes and up the dunes, and I want to go right and I want to go left, and I have absolutely no idea. To this day, I know what's going on behind the scenes. It's an indirect thing. But really, in terms of feedback, you don't need the dune buggy. The person at the bottom here that I was mentioning is looking at a video. It's probably a kid's movie. And if the person is doing the right thing with her brain, the movie just plays on and it's an enjoyable thing. I'm sitting down and watching the movie. If you lose attention or if your brain's not doing the right thing, the movie starts being chunky, might stop going forward, might start rewinding. Things that is bad feedback. Your brain doesn't want that. I want to watch the movie. So the brain learns how to do the right thing. The guy at the top, that's mostly how neurofeedback is done nowadays. Instead of having a visual cue, you basically get a sound, okay? Either it's nice music that gets interrupted if you're not doing the right thing, or you basically get a ping every time your brain has achieved one level of things. And your brain wants to hear those pings. Oh, that's a good one. And if you're not doing the right thing, it goes, eh. You don't want to hear that one. So something as simple as you want to get as many pings in an hour as you can. All you need. And the brain starts figuring out how to optimize the number of pings. And what you're really doing is you're training the brain to adopt a neural network that it doesn't do naturally. Kind of learning how to ride a bike. Okay? It's amazing how that works. It's simple. It's very difficult to explain why it does that, but it is the same process as learning to drive a bike. And once you've learned balance, you now have the neural pathway available to you. So people can get off Ritalin and learn how to control ADD by training the brain. Is this done quickly? It depends on how severe your situation is, okay? But it targets the whole brain circuitry at the same time. It's individualized. So you saw the 12 networks. After I'm done with the Navy, I'll be able to personalize a neurofeedback for a midshipman that's great at 80% of the stuff, sucks at these 20%. We'll be able to correct that as opposed to ask the person to move out of the Navy because they're not good, which is expensive to the US taxpayer, okay? And it's not invasive. Now, the brain will not do anything that's bad to the brain. So it's not, you're not going to get a person to self-inflict an epileptic seizure unless you're already epileptic, okay? It's programmed not to do that. So it is, in fact, non-invasive. It's drug-free. There's no Ritalin. You don't have to. In fact, we don't want that. We want your brain in its normal condition. And after a couple of weeks, I can do another brain map and see, has your brain changed? Okay, and depending on which technology you use, and we are going to use some very high sophisticated Z-score neurofeedback machine that's pretty sophisticated from a software perspective, okay, we'll be able to assess how good you are, and in fact, which network should we concentrate on next as part of your training. So here we go. You've learned a lot about neuroscience. Let me just finish off on organizational neuroscience and some of the lessons that I've learned. I'll go through this pretty quickly. Examples of what I call impactful organizational neuroscience research, and there are not that many. As I said, there's 500 people working on this. We may see maybe a dozen decent papers a year. Maybe not. Okay, but it's growing. People are now figuring out that it's not impossible to do this work. I really like things dealing with leadership, either adaptability How can you do dynamic decision making? You're interested in ethical leadership. It's a growing domain just by itself. Okay, you could have a full career on the neuroscience of ethics and the ethics of neuroscience. Okay, believe it or not. You saw that we were involved in the first team to do team neurodynamics. Nobody's done any work on team neurodynamics except for the people that were on that original team. There's some work at UCLA, but we were working with UCLA, so they went with it. So anything that deals with collective intelligence would be fascinating to look at. Hasn't been done. Anything that deals in the work environment with stress, cognitive load. Okay, it's not just a helicopter pilot, okay, and their telemetry. It'd be good, okay, to know where you are and are you ready to make that decision or do that work today. Executive performance, that's where I guess the money is. Maybe at some point I'll see some of it. So far it hasn't happened, but it might, because I think there's a lot to be said about executive performance. I'm very interested, and I'm working with people in Munich, Germany on entrepreneurial cognition. By the way, I mentioned San Diego as a seat for a lot of the technology. Oddly enough, Bavaria, several companies. Okay, the cap that I use right now is produced in Munich and also in San Diego. Oddly enough, the Americans in San Diego got the rights to produce for the American market. But they do a lot of work with entrepreneurs. Okay, at the Technical University of Munich, I was visiting professor there, and we got going on studying entrepreneurs, and that's fascinating because the brain of a true entrepreneur is different than most people walking the street. Organizational learning, cognitive transformation within organizations would be fun, and neurophysiology of strategic thinking. I'm doing some work, although it's now taking somewhat of a back seat, on dynamic capability, the work that David Teese has done at Berkeley. I've worked with David trying to figure out how we can do the study of the brain of people that do dynamic capability. Now, unfortunately, I see a lot of this that I call garbage research. Okay, brain region hunting. Okay, and I can see myself in this. Okay, leadership activates the prefrontal cortex. The answer to that is duh. Okay, there's nothing there, and a lot of people think that that's original research. It's not. Neuroscience that doesn't have any organizational relevance. Okay, don't send a study to the Academy of Management with an N of 3. It's not going to get published. Okay, might get published in a medical journal if it's something good, but typically it's not that good. And there's a lot of reverse inference overreach. In fact, one of my co-authors, I remind them all the time that, yeah, the default mode network has something to do with it, but the default mode network is 60% of your brain. I would hope it has something to do with it. Okay, but the claims such as this EEG pattern proves the leader is ethical, probably BS. Okay, it might contribute, it might be a precursor to or whatever. So brain activation by itself doesn't demonstrate anything. Okay, it's a biological measure, and you have to be careful in how you say that. This person is a great leader because they've got great alpha symmetry in the frontal part of the brain. Really? Okay. So I see a lot of that, and here more. Okay, technology led. Okay, my paper is great because I talked about the EEG system. No, a lot of reductionist stuff. Okay, you take a measure, and because of that, you explain the whole history of mankind. I've seen a lot of papers that attempt to do that. That's not good. A lot of small sampling, and what I introduced earlier on as either neurojournalism or neuropabble. Okay, people that just look. If you talk about the brain, people are going to take a look at it. Ongoing issues that I see. I would like to see more work that really studies organizations, and more organizations are multi-level. Okay, you got teams, you got systems, you got strategies, you've got organizational culture. I've done a lot of work on culture, but never with a brain perspective. Okay, static trait neuroscience. Again, there's the classic: were you born or were you made? There's a lot of people that take a look at fixed indicators. of leadership, but they don't particularly take a look at context of leadership. The Army is one of those, okay, place where the culture is more important than an individual leader's capacity. And there's a lot of studies that I see where the methodology is very sophisticated, but it's applied to a very underdeveloped theory. And there you go. In general, if you address a meaningful organizational phenomena and you're grounded in decent theory, both neuroscience theory, but also organizational theory, and you use the neuroscience appropriately, like my very first example of, showing that something is semantic versus procedural, vice versa, and you do attempt to integrate multiple levels of analysis, or at least acknowledge them, and you create insights. And those insights, they might not be available to you if you didn't use neuroscience. And hopefully some of you are going, could that work in my discipline? This was just me sitting down and trying to figure out, where can I apply this stuff? And I think there isn't a paper that's been written over the last 100 years that we couldn't somehow figure out a mechanism to use neuroscience to educate us more about that subject matter, whether it's economics or strategy, entrepreneurship, OB, marketing. Okay, there's a lot of neuromarketing stuff, neuroeconomics. My friend Paul Zach in California has done a lot of work in that area, decision-making, finance, corporate social responsibility, ethics, HR stuff, communication, and so on and so forth. And I'm sure if we sat down for 15 minutes, we would double and triple the list. So I gave you all a grand total of 9 minutes, okay, for our session to be actually over, but hopefully some of you have got questions, and I'm more than happy to stick around beyond this. And if you got my emails there, feel free to send me something. I'll be more than happy to answer those questions for you. Yes, sir.
Thank you, Pierre. This is probably the most fascinating research presentation I've heard in a while. So thank you so much.
Good.
The question I have: how do I get started if I want to do this type of research?
Look, first of all, it helps if you've got an environment where somebody has access to the machinery. Okay, it's not as expensive as you think it is. You can set yourself up for, I'll give you an estimate of less than $20,000 to do the studies at the sophistication that I showed today. Clearly, you can also go out and buy one cap at 30 grand, 35 grand. I wouldn't suggest it. Then it's your time and effort. It's always good to get in your family somebody that's a neuroscientist. That it's more difficult than you think to find somebody that won't just blow you off. When I first started doing this, I spoke to a variety of neuroscientists that just looked at me as a quack. And then I met one of the top EEG neuroscientists in the world, a guy by the name of Bob Thatcher in Florida. And he was amazed. He said, I was expecting a guy like you to show up here some 20 years ago. And I'm going, what are you talking about? He said, this is ripe for exactly the type of stuff you want to do. And I asked him a question, and he answered it completely differently than everybody else. I said, do you think I can use EEG to help me develop better leaders? And his answer was, it depends. I was listening. He says, tell me about leadership. So here was a neuroscientist, okay, of incredible reputation, curious to understand what is the concept of leadership and what are the variables. How do you go about figuring out if somebody's good or not? If you can figure out that this set of people are very good at this and these people suck at it, then you're in good shape to figure out, is there a relationship between the brain and what makes a person good or bad? So he was much more interested in my science, as much, in fact, as I was interested in his. That's tough to find. Nowadays, it's easier because there's a lot of individuals that are recent management PhD students that now have a mechanism through the academy and whatever to get into a legitimate, 30 years ago, I'm a nut. Don't, I've been told by a dean, don't quit your day job. Okay. 3 weeks later, I'm on the front page of the Wall Street Journal. Okay. So I knew there was something there, but okay. You need to get somebody in your environment that can actually coach you to go, to use the system. But it's a software system. Okay, I've been using a simulation for the last 2 years. Okay, business strategy. I never used a simulation before. I'm pretty good at it now, but it took me 1.5 years to get to the point. So it's the same thing. So once you learn the software, and the people that develop the software are very helpful. They don't deal with people like me very often. They deal with neurotherapists, people that do ADHD and ADD. I've put caps on about 2,000 people in my life, plus or minus. Okay, so it's relatively cheap, but you do need the base. You need to get people that are going to give you the time and effort to help you out. Any other questions, guys? Anything?
Somebody else.
That marketing example was so interesting for me. Like, and also like even the you just show one word and then we think about so many other things.
Yeah.
That's also like in marketing right now, let's say when you create content, how you create the clickbait, like just whatever you call title the video, you just you choose the video, right? Or let's say Google Ads, whatever. Sure. Like that one word it's very important and it's can make or break you, So
correct.
That's a fascinating connection between that one word.
Yeah.
And
by the way, you just defined what could be an organizational neuroscience study right there.
Mm-hmm.
Okay, of trying to figure out what word and show it, from a brain perspective, what response.
My thing is like, I know this stuff. Like, I know that this word is a clickbait. I still cannot stop myself clicking that video. It's just such an interesting thing that such a powerful connection with the brain, or however we are programmed, that we still click on that,
And somebody that might be sitting right next to you wouldn't care less about it.
There's a fascinating study by Sendil Mulianathan from University of Chicago.
Yeah.
It's called scarcity, right?
Yeah.
And what he looks at is people who are, looking at meeting their ends, right? Yeah. And when you ask them a set of words and how much they remember, all the words that they remember are related to the scarcity and their life condition, while others will remember other words. So
I don't know that study per se. I've read about probably a dozen studies that are similar in scope. None of this surprises me. It's trying to make conscious what we've done subconsciously for years. Okay, it took somebody to figure out what would be the research question that I could learn something about this phenomenon. That's what's interesting in organizational neuroscience, is everything you look at, we probably could design 10 different studies today that were all fascinating. My first one was an award winner, and all I wanted to say is, could I predict somebody's MLQ score using EEG? That was it. And the answer was not only yes, it was hell yes. And I've been trying to redo it, for the last 15 years because I know that it's going to work. And I'm happy that the Navy thinks I'm also capable of doing it again. So, by the way, in the meantime, we also developed something called the Brain Performance Index, or the Brain Function Index, which actually is more precise than a HiQ at figuring out how effective and efficient your brain is. The HiQ, if you ask, people that do a lot of neuropsychology, are going to tell you it's an imprecise variable. It's a surrogate. Okay, and we all understand IQ and whatever, but there are better measures of who's got the goods and who doesn't. If you introduce neuroscience in it. Yeah, go ahead.
So people on
the brain activity while one is sleeping or staying inactive.
Actually, in my case, I've had colleagues that do sleep and also sleep deprivation. In my case, I do the brain at rest
At rest.
but you are active, you are alert, but at rest.
Oh, okay.
Often a person might be sitting down in the chair and I say, hey, just sit down, close your eyes, don't sleep, don't meditate. The person that meditates is a person that learned how to slow down the the speed of your amplitude in the front parts of your brain. So they go from beta to alpha. And okay. By the way, that's how, many years I worked on a DARPA grant. DARPA being the high-end intelligence research in this country. And it was fascinating being in the same group as they were teaching people how to become sharpshooters. Okay. And it's really an exercise on doing a very special type of meditation where they actually control their brain waves. So at the beginning, they put caps on and they get feedback on what their brain is until they learn what you need to do. And then they do it without getting the feedback. They just know, okay, I'm now in a position, I have to control my breathing and whatever. And they slow down their brain movements, okay, in the frontal part of the brain, and it increases their accuracy incredibly. And some of these sharpshooters can shoot a bullet a mile away and hit bullseyes.
Interesting.
Yeah. Same thing as when they want to find. You would think that if you were looking at a satellite picture of where there might be missiles pointed towards us, and my research has been military, so I've seen a lot of these things. You would think they just look at a big map and they just look at the whole map and say, oh, there. No, that's not what they do. They've actually taken the map and they, just like voxels, they create small little squares of the map, and they've trained some specialists to take a look at that square and say, is there something dangerous or potentially dangerous in that little square? So instead of looking at the whole map and just scanning like that, they get a precision of X. They will actually go through this process where this guy's looking at the map like 1,000 pieces at a time, and they put yes where there's some danger. They're significantly more precise when they look at the map in pieces than the whole map as a whole, like 10 times more precise.
So they focus on the areas that
They focus on one small part of the map, one at a time, and they do the whole map like this, as opposed to taking a look at the whole map and trying to figure out where the danger is. They really concentrate on one thing at a time, but over and over and over again. And the brain is so good at learning this, it becomes automatic. So they just go for, okay, the whole map in pieces, and they are 10 times more, maybe I'm exaggerating, but several times more precise at figuring out the danger where there's a high probability of having a missile pointed towards you than looking at the whole satellite picture as a whole. Who knew? Somebody came up with the research methodology and they've trained these people. So we've got experts in the military with all of this expertise. I like the concept of hypervigilance. Okay, people that get trained, they're sitting down in a Jeep or in a Humvee, and they want to smell or take a look at the environment and say, "Whoop, there's an improvised explosive device right there." So you're driving and there's somebody sitting down in the front seat, and you just think they're sitting there on a ride. No, they're scanning the horizon and they're trained to identify where there might be an explosive device. We've got some people in our military that they're the biggest consumer of neuroscience research, from my perspective. I've seen some very fascinating things.
Yeah, my question was relatively minor.
Mhm.
Then somebody's at rest and the brain is still working. I think
Yep.
that might be a function of what they had been doing up until then, because you need to have some material to
Yeah.
have the brain work with.
Yeah.
And unless you've been sitting idle forever.
Yeah. Look, your brain switches very quickly from being on task to not being on task. And when you are at rest, whether it's 2 minutes, okay, you might have been in this lecture today, and at some point you're kind of daydreaming, and then you go back, oh, let me listen to Pierre a little bit more. Your brain does this a million times, okay, a day, okay, all the time. And when you're not entirely concentrating on something, it's doing what it wants to do. So when we say at rest, we want the brain to be alert. So that means if a tiger walks in the room, we want you to react, there's a tiger in the room. But at the same time, we just want you to do whatever, but nothing systematic. Don't count by 3 until you get to 1,000,000. That's not good. That's a profile. What we usually tell people to do is sit down and think of your happy place. Where do you want to go on vacation? Why? Who would you bring on vacation? So they're sitting there and you can see them, then they okay, have a little smile on their face, and they're just, that's where you want to be. That's when daydreaming is fine. Meditation, not good. Being on drugs, not good. We always ask our subjects, if you use some form of psychotropic drug over the last 24 hours, it is amazing the number of people that are on all types of prescription drugs to control the human brain. That's why I like that military. None of them are. Some of them should be, but they're not. I should watch it. It's recorded. I shouldn't say that. They're all perfect. Yeah.
What about emotions? How does it affect,
It's everything. Now, emotions are very important. And a lot of my research, deals with emotions. For example, what's right or wrong in ethics? The right or wrong is your emotional part, because you can't do that because your brain won't allow you to do that because it's not right. That's emotional. Okay. There's actually many parts of the brain that are studied. Okay. The cognitive aspect is big. The emotional aspect is big. But there are other aspects that are well understood, but not well studied. The concept of conation, is fascinating to me. You have two people that are equally intelligent, that are very similar in terms of their emotions and whatever, and one might be motivated to do something and the other person might not. So it's not the cognition that explains it. It's not the emotion. There's something else there. Take a look at conation in the dictionary. The conative ability of people is an understudied component of the human brain. Back there, yeah.
Hello, my name is Budak. In the context of organizational neuroscience, what can you tell us about telekinesis and mirror neurons?
I'm not a big expert. I know what I know, and I reminded everybody I'm not a neuroscientist. I'm basically an industrial engineer that deals with a lot of numbers. There are a variety of mechanisms to get your brain to do things for you. And in terms of how you think, how you communicate, and I think that would be fascinating to do a study of nonverbal communication of people around a table. One of the most interesting studies that I never did, I was this close of doing for about 3 years. I was a dean at a Franciscan university with friars on campus. And I was fascinated with what would motivate a normal human being to become a friar. Okay. And I in fact had a study, and I had New York approved, and I was approved to do a study on the brain of people that were friars. And the study had the code name Wire the Friar. And it was fascinating, but we never got, religion, meditation, communication, how the brain works and how we interconnect each other to have it's all fascinating to me, but it's not what I do.
Say nonverbal communication, what do you mean, like the body language?
Body language is one thing that assumes visual. Okay. Why is it that a husband and wife married for 30 years can be at the breakfast table, each doing their thing on their phone and whatever, and they're having a conversation? I think that's fascinating. Okay. And what is it? And you ask a person, did you speak to your husband about this? Oh yeah, we talked about this morning. And they would both agree they talked about it this morning, but if you had a video, there was no conversation. How did this occur? And that might be somewhat related to what you're talking about, but I think all of this is fascinating. Okay, and that's because it's part of that 95% that we don't understand, which is the subconscious. Okay, our brains do a variety
Or
of.
sometimes like you're talking, probably something happens. And
Yeah.
You just think about.
How about a situation where, you're in a part of the world that you've never been before, but you're sitting on a street corner or whatever and say, I think I've been here before. It's freaky. And who knows, maybe you've been there before. That's why I think in the next 20 years, there's going to be more and more neuroscience based research. By the way, the most out in left field person that has ever talked to me says, what you're talking about is fascinating, but it's only a transitional research. Okay. What do you mean? And he said, it's not about the brain, it's about your DNA. So there are some people that study the human genome that believe it's not about the brain. We can actually create the brain that we want as long as we understand what the code is. So they look at neuroscience as being just, it's right now it's the end thing, but 50 years from now, it's all going to be about, there are some people in the lab, by the way, and Arizona State, I know of a couple of studies like this where they will plug a computer chip into your brain. And, right now what they do is mostly for mobility, okay, getting an artificial limb to work, but it could also be for language. Imagine a day when you say, oh, I have to go to Turkey. Okay, so I'm going to put my Turkish chip in. So you get off the plane and you're home. Okay, it's wild, but it's not as wild as it was 50 years ago. Yes, sir.
And you said brains switch very quickly between resting mode to active mode.
Yep.
The time to switch resting mode to be going active mode, is it a dimension of the intelligence? Is there a significant difference between
Is there a
humans?
relation? Yes, I think. You see, the true neuroscientist would say something like, what do you use to measure intelligence? Okay. In part, what I mentioned was IQ is an imprecise measure of intelligence, and neuroscience use their own version. The neuroscience use of efficiency and effectiveness of the brain would say absolutely there's a relationship between the reactionary time of your brain and efficiency and effectiveness of the brain. I am not convinced our social science definition of the Wechsler IQ test is going to capture that level, but it might. I would think that yes. Okay. There is something called flicker fusion. Okay, how quickly your brain reacts to certain events, how fast are you, and we're all kind of wired slightly differently, is in fact has been shown to be a correlate of intelligence.
And my last question.
Sure.
When people spend time together, we think alike.
Yeah.
And you will talk about voxels in the brains. Yeah. Is it the stimuli that impact brains of the people who spend time together, or is it something else?
No, it's the same thing as this couple that's been married for 30 years and can think each other's sentences. Your brain is always learning, consciously and subconsciously. Okay, just like me learning how to drive the dune buggy I still don't know what I did, but I can drive the dune buggy. Okay, that's very subconscious. So if you're in a group and you're together for a while, and that's, at some point, I'll probably go back to my roots and do a study of virtual teams. If you're working alongside a colleague on a project for 2 years, either you like or dislike these people, but you're much more alike from how you think about this project than you were when you started the project. Now, if you're working with the same team, but virtually, there's a computer interface between you, you probably don't have as much of a brain connection because it's moderated through the technology. But yeah, sitting next to a person and working alongside people, you become comfortable, you understand how they do things, you react to them. It's a difference, okay? Right now we've got the Knicks, okay, playing. Are they a better team than the other guys? We don't know, but the real teams are those where, believe it or not, the brains got together and they're working together towards something. They can bet with you, if we did a neuroscience study of professional sports teams. By the way, Ronaldo was in town yesterday. Did you know that? In Florham Park. Yeah, yeah. The Brazilian team is staying at the Hyatt in Morristown. I know there's a big police presence. Anyway, sports. When teams work together for a long time, they actually get a personality that's the team's personality. Yeah. We call it chemistry, but your brain is also part of that chemistry. Absolutely. And you could study it neuroscientifically, and it's there. Yeah.
Should we give Pierre a round of applause?
I appreciate it. I'm really happy I've done this. I was looking forward to this, and I thank you for the invitation, Omer. I'm very happy I've done this.